Morning, everybody. Hope we're doing well. We're going to continue with our mitosis, meiosis, cell division, cell growth unit. Yesterday, we went over cell division, we went over interphase, and we kind of discussed why it's important for cells to divide instead of just continuously growing. So what we're going to do today is we're going to actually talk about mitosis itself. We're going to go through each stage of mitosis. There are multiple, and we're going to discuss what happens in each stage and how you can tell by looking at a picture of a cell what stage it's in. So there are a couple of things to do today, but they all relate back to what we did. So make sure you review yesterday's work if you need to. Let's get started. So that's what we're going to do today. So yesterday, let's just do a quick review. We spoke about interphase, right? Remember, interphase encompasses these three phases, G1, S, and G2. G1, the cell doubles, and then the organelles double. S is where DNA replication occurs. And then G2 is where special structures are formed in order for the cell to actually divide. So this is all known, again, as interphase. Interphase is part one, or rather step one, of mitosis, right? So let's take a look at that again. So interphase, right? So this accounts for 90% of the entire cell cycle. Remember, go back to this part here, G1, S, G2. This is the majority of the cell cycle. So in reality, it actually takes up 90% of the entire time it takes for a cell to split. What you need to remember about interphase is what we learned yesterday, right? These things here, G1, S, G2. But to go further into it, in order to identify it, because the regions, what they might do is they may give you a picture of a cell, and they may ask you what phase that cell is in. And throughout each phase, there are a lot of different ways to figure out what phase it's in. I'm going to just tell you the most common ones. For interphase, everything is intact, right? The nucleus is well defined. You can see that it's just a circle. You could see that it has a membrane around it, the nuclear membrane. And then outside of the nucleus, there are two centrioles, right? These yellow things right here are centrioles. Basically, centrioles are organelles that help function to pull the cell apart, right? They will begin to move apart, and then that helps the cell split. So during this time here, they're not moving apart because the cell isn't splitting yet, right? It's still through interphase. It's still growing. It's still replicating the DNA that's in the nucleus. So nothing is splitting, so it doesn't need to move. It's kind of just sitting there. So if you see a picture where the nucleus is intact and then the centrioles up top here are together, then you know you're in interphase. Because again, nothing has happened yet. It's still during the growth phase. So another couple of things to remember about interphase are, again, we're just going to review what we did before. So the G1 phase, which we know as the growth one, this is where the cell doubles in size. This is where there's a lot of intense biochemical activity. So a lot of stuff's happening in the cell that's making it replicate the DNA, that's making it grow, it's copying the organelles, it's doing all that stuff, right? So we see the cell doubles in size, and the enzymes, the organelles, and other molecules also double, right? That's G1. Now, S again, remember, is where the chromosomes duplicate, DNA duplication. And they're replicating, they're duplicating, but they're kind of just a mess. Like if you look in this picture, it's all fibrous. They're not really structures yet, but they are there and they are doubled. And then G2, again, is where we assemble our special structure. So for interphase, what you need to remember again is the three phases that occur within interphase, right? G1, S, G2, what occurs in each of those, and then how to recognize interphase. And again, we recognize interphase by having an intact nucleus and centrioles that haven't split yet. All right. There are a couple other things that we see here. Nuclear membrane, again, that's just a membrane around the nucleus. So we learned about cell membrane when we talked about organelles. Cell membrane is the membrane around the entire cell. Nuclear membrane is the membrane that's around the nucleus. And then nucleolus is this tiny little circle here. It just means that it's a little structure within the nucleus that helps the DNA, helps all of that stuff form. That's not super important. It will disappear eventually throughout the other phases. So just take note of that as well. All right. So that's interphase. That's phase 1. Now, again, repeated because this is important. Interphase includes G1S, G2, and this is what occurs before mitosis actually begins. Remember, keep going back to this here. This is a really good diagram for you to remember. G1S, G2, all three of these together are interphase, and then mitosis itself will start. Sorry about that, y'all. Let me mute my phone. Mitosis itself will start, right? So when we get to mitosis itself, mitosis has 4 phases of its own, right? Prophase, metaphase, anaphase, and telophase. So those are 4 separate stages. So if we were to look back at this diagram, really what we have is interphase, right? Then you have prophase, metaphase, anaphase, telophase, which is all mitosis, and then cytokinesis. So very simply, you could talk about the cell cycle and you could say that it's a couple of different processes, but they all end up being the same thing. We're going to talk about that at the end. But let's get into these phases. So we've concluded interphase, we finished the growth, we finished the DNA replication. Now it's actually time to split, so we have to get into mitosis. So the first phase for mitosis is prophase, right? So there are, sorry about that. There are 2 parts of prophase. There's early prophase, and then there is late prophase. So we're going to talk about both, right? So in early prophase, chromosomes will start to coil and thicken, and they become distinct. So now if you compare this picture to what we looked at in interphase, it's very, very different, right? You can see the nucleus is still intact, the membrane is still there, but the DNA is now defined. You can actually see strands of DNA as opposed to in this picture where they're all kind of just coiled up and fibrous. It kind of looks like spaghetti, right? So if you come back, you can see that it's more condensed, right? Also notice the nucleolus that we spoke about, the little circle inside the nucleus is gone, right? That disappears. And, there are a couple of other distinctions. One other important thing to remember is the centrioles here have now moved. So remember, in interphase, there is no growth. There is no division happening. But as we start prophase, the division starts to happen. So the centrioles move, right? And you see that it forms these things here called microtubules, right? That's important to remember. Microtubules are kind of just, how can I describe them? They're like fibrous little strings, you can think of them, that come off of the centrioles, and they just grow as the centrioles split, right? It grows, and they kind of keep both centrioles 1 and 2 attached through the microtubules, right? So this is prophase, early prophase, okay? So again, early prophase, the stuff to remember is very simple. DNA starts to form and it becomes condensed. It looks more structured. Centrioles will start to move away from each other. All right. Now, when we get to late prophase, you see now the picture looks very different, right? There are a couple of different things that happen in late prophase. So the first thing that you have to remember is the nuclear membrane that we spoke about, the membrane that covers the nucleus, fragments. It breaks apart, and it kind of disappears in a way, right? You see that there really isn't a nucleus anymore, right? If you look at this picture, it's very, very well defined. You could see the nucleus here. But when you get to late prophase, the nucleus fragments and it becomes difficult to see, right? It really is not there anymore. The spindle, right, which we learned before, is what is forming from the centrioles. Remember, we have here, this is the microtubule that's starting to make the spindle, right? It's separating, the centrioles are separating. These microtubules in the middle are being made, and they're starting to form. Once we get to late prophase, it has formed completely, and you now see that one centriole is at the top of the cell, and one centriole is at the bottom of the cell, and the microtubules go right through the middle, right? So this right here is a key way of determining what phase you're in. If you see the centrioles at the top and bottom poles of the cell, and you see that the microtubules go through the middle, you are in late prophase. All right. So what else do we notice about the microtubules? Well, we notice that they are going through these chromosomes now, right? They're going right through them. They are attaching to them, which is important. And we're going to talk about why that happens in a second. But just know that the spindle, right, which are these fibers that go through the middle, they help separate chromosomes, right? Because remember, the entire end goal of this process is to replicate your DNA and to split a cell in half, right? You are splitting a cell. That's what mitosis is. So if you have DNA here, right, you have 1 and 2. These are chromatids, right? Make sure you know that this entire thing here is a chromosome, but this one top is a chromatid and this one is a chromatid. 2 chromatids equal 1 chromosome. So we need to split them in half because the new cell that we're creating needs to have equal numbers of chromosomes. So you have to cut those in half. So the spindle will eventually separate those chromatids in half. All right. So that's late prophase. And that's basically prophase. You know, we have early and late. Prophase is the most complicated stage in mitosis. So it gets a little more simplistic from here. But these are 2, prophase, early and late. All right, make sure you go back, listen to this again, review it, ask questions if you need to. Please feel free to post or email me. I know that this can be confusing, especially since we're doing it like this. So I'm going to be available for this particular. I'm going to be available for everything, but for this one, please make sure you don't hesitate to reach out if you need any clarification. All right. So after prophase, we move to metaphase. Now, metaphase is very simple. Metaphase is an important step, and it's real easy to identify. Again, the centrioles are still at the opposite sides of the poles of the cell, but the main difference and the way that you can identify metaphase is that the chromosomes line up at the center, right? They are all lined up right in the middle of the cell, all of them. Okay? That is the identifying factor for metaphase, is that they all line up at the center of the cell. And that's really it. So during metaphase, it transitions from being kind of this mess here, where the spindles and the microtubules are through the center, but it's kind of still not really formed anything yet, to in metaphase, you now have a very defined cell where the chromatids are along the center of the cell, and they are attached very distinctly. All right, so those are metaphase. Real simple. All right. Anaphase is also very distinctive. This is an important part of mitosis, and it has another distinctive characteristic. You should be able to recognize what it is before I even tell you. The centromeres divide, and the chromatids start moving to opposite sides of the cell. So now these chromosomes and chromatids that used to be lined up in the center, right here in metaphase, right? The microtubules that we spoke about, remember I told you those are kind of like strings that come off of the centriole. They use to attach to the chromosome. And their job eventually is to pull them apart, like I mentioned before, so that you can have them in each new cell. And that's what starts to happen in anaphase. You see that the microtubules that are attached to the top chromatid, to the top chromosome centriole, to the top centriole starts pulling apart the top chromatid, and it starts pulling it that way. And at the same time, the bottom centriole is pulling the bottom chromatid, and it starts pulling it down this way. And this happens for every single chromosome and chromatid that are found in that cell, right? And the microtubules can actually shorten, and that's how they pull them back toward the top or back toward the bottom, right? So at the end of anaphase, the two ends of the cell will have equivalent and complete sets of chromosomes. They will all be pulled backwards. They'll all be pulled back. And anaphase is over once they all reach their respective sides of the cell. All right, so the way to determine if you're in anaphase is if you see the chromosomes and chromatids still kind of in the center, but they're no longer attached. They're now being pulled apart. That's how we're in anaphase. Now, telophase is also an extremely distinctive phase, right? You're going to see that, hopefully you're seeing that each phase looks different and each phase has a different characteristic. So it shouldn't be super confusing, right? You're very unlikely to get confused by a phase if you remember the characteristics, because they really don't look alike, right? So telophase. What we have here is you've seen now that up until this point, right, there is no nucleus anymore. There's no membrane. That disappeared after metaphase. In telophase, the membrane begins to reform, right? The nucleolus will return, which disappeared as well, the little thing inside the nucleus. And then the cell begins to pinch in. So this is called a cleavage furrow. Please remember that name. The cleavage furrow is the point where it pinches. So realize this is still one cell, right? Technically, still one cell. The only thing now is we have the one centriole at the top, the one centriole at the bottom, like we did, right? We still had that here, one here, one here. And now we have chromosomes and DNA at one bottom end, chromosomes and DNA at one top end, which is what we have here, right? Once they reach their respective sides. The only difference is now in telophase, the cells are starting to form their own, right? So the membrane begins to form again. The nucleolus begins to form again on both sides. And then this cleavage furrow is going to form. So think of the cleavage furrow simply as where the pinch in is. So if you were to take this cell here and you were to squeeze it on both sides, I know you can't really see what I'm doing, but if you were to squeeze it right here and squeeze it right here, you would end up in telophase, looking just like this. So the cleavage furrow is that pinch point, and telophase is it looks like this. All right. So the end result of telophase is two cells that are exact copies of each other. This eventually will turn into two cells. What's going to happen is this cleavage furrow is going to keep pinching inward until it splits. And that's the next phase that we're going to look at, right? This, though, however, is the end of mitosis, right? So telophase ends off mitosis, okay? And that's really it, right? So I'm going to quickly run through each phase one more time, but I'm going to just be quick with it, and I'm going to just get you the points that you need, just kind of as a recap, all right? So we have interphase, right? Interphase is G1S, G2. During interphase, we have the cell doubling in size and the organelles doubling during G1. We have during S phase DNA replication, and we have during G2, we have a special assembly of structures that will eventually help the cell split. And we now know that those special structures are the centrioles, right? So that is interphase. Now, once interphase is complete, mitosis can begin. Mitosis is prophase, metaphase, anaphase, and telophase. Where are we? Yes. So prophase, metaphase, anaphase, telophase. So after interphase, we're going to jump right into prophase. Early prophase is when the centrioles start moving away from each other, microtubules start to form, and the spindle starts to form. Realize that in early prophase, the nucleus is still intact, but the difference between this and interphase is that the chromosomes are a little more distinct. Okay? Late prophase, the centrioles have moved to opposite sides of the cell. One is on the top, one is on the bottom. The spindle has formed throughout the center. It's starting to attach to the chromosomes, and the nuclear membrane has disappeared. There's also no nucleolus. Okay? During metaphase, the chromosomes have lined up in the center of the cell. Right? Each chromatid has attached to its other chromatid to make a chromosome, and they have lined up at the center of the cell, right down the middle. Metaphase. Anaphase. The microtubules start to shorten, and they are pulled upward and downward. And when they are pulled, because they're being shortened, they are ripping apart this chromosome, and they are each bringing up a piece to their respective side. So the chromosomes in the middle will break, and they begin to pull the chromatids backwards and forwards, depending on which side they're going to. Anaphase. Right? Telophase. The chromosomes are now on either side of the cell. The membrane of the nucleus starts to reform. The nucleolus right here and right here start to reform. A cleavage furrow is created, which means the cell is pinching in on itself, and it eventually creates two distinct cells that are exact copies of one another, and that is telophase right there. And that is the end of mitosis, right? Now, it's not the end of the cell cycle, because remember, if we go back to our picture here, there's still one more part, cytokinesis. So we're not done with the cell cycle, but we are done with mitosis. Okay? We're not going to do cytokinesis today. We're going to talk about that and some other stuff tomorrow. But we are done with mitosis. We're done with interphase. Remember, we're done with interphase. And then we're done with prophase, metaphase, anaphase, and telophase, which is mitosis. Okay? So this, normally what I would have done in class is I would have had us work on this and try and determine what the numbers are. These are actual cells that are that were taken microscopically, and I would have had us try and figure out what they are. You can see though, like if you look at this one here, number 3, right? There's nothing in the center. It kind of looks like things are being pulled apart, and that's because it's in anaphase, right? It's this one right here. Where are we? Here. See, it kind of looks like it's being pulled apart. So if you were to look at this one, you would see that it's anaphase. This is metaphase because it's kind of lined up in the middle, as is this one. It kind of looks like there's a line right down the middle. Metaphase. A way to, again, I didn't mention before, meta. You could think meta, middle, both start with M. So that's how you know it's in the middle. Prophase. These are all prophase because these just don't really look like anything. They're kind of just, you know, circles with DNA in it. And then telophase, you could see very clearly a telophase right here, and very clearly a telophase right here. There are two separate cells, right? Yeah. So we are going to do cytokinesis and a couple of other things tomorrow. Um, we're going to finish this lesson right now. My recommendation is go through this video again. Make sure you know the differences between each phase. So make sure you know the names of the phases because you have to know them. You're going to have to know how to identify them. And you're going to need to know what order they go in. So make sure you know the names. Make sure you know what occurs in each phase. And make sure you have an idea of how to identify them, either through a picture or through a description, right? And a simple question could be, during this phase, the chromatids line up in the center of the cell. What phase are you in, right? And you'd have to know that that's metaphase, right? So make sure you review that. We're going to have an activity that I'm going to post along with this. That'll be your task for today, which will help you remember that stuff and identify it. So you'll be all right. Again, as always, if you have any questions, make sure you let me know. Post here, email me, email Miss Steele, whatever you got to do. Just make sure you get that done if you have to. All right, guys, make sure you're checking your Google Classroom and schedule it every day. And I will see you tomorrow.